name is Maria Rangel, and this summer I did work under the Department of Psychological and Brain Sciences under the mentorship of Sierra Webb in the Salimsky Lab. And my project involved understanding the role of myelin basic protein in the working memory. So to begin with, human cocaine addicts and preclinical animals who have self-administered cocaine have shown a deficit in myelin basic protein and also their working memory. And we can refer to working memory as short-term memory. And the myelin basic protein, so here on the left I have a picture of an axon, and what myelin basic protein has to do with this is that here surrounding the axon is the myelin sheath, and which consists of the myelin basic protein. And the myelin sheath is important for neural action potential propagation. And here on the right, I have a cross-sectional view of a rat's brain, and we're going to be focusing in this area, which is the ventromedial prefrontal cortex, which is responsible for working memory. So the approach, the way we ran the experiment is we micro-injected a virus that simulates what cocaine would do to these rats. And then we waited for at least 30 days, and then we trained these rats and then performed 2 tests that tested for their memory. So the subjects, as I mentioned, is these lovely rats, and the tests were performed in an open field box, as can be shown in the middle. And then we had 4 different objects, and we used almond and lemon extract. So the first test that we performed was the novel object test. So before we put these rats, before we performed these tests, we allowed the rats to habituate for a total of 5 minutes so they can get used to the environment. And then we performed the familiarization phase, where we introduced 2 of the same objects and then added the same scent. So for example, here on the picture on the left, we would add the 2 identical objects with, let's say, lemon scent. And then we put the rats back in their cage, waited for 90 minutes, and then performed the exploration test, where we introduced a complete new object and with a different scent. So we would add lemon scent. And I get asked a lot if the rats have a preference for lemon or almond extract, and there has been some studies that there isn't, so we don't have to worry about that factor. The second test that we performed was the object in place test, where we allowed the rats to habituate for 5 minutes, and then we put them in the box where we introduced 4 different objects, and then we put them back in their cage, waited for a total of 90 minutes, 90 minutes, and then we did the exploration test, where we switched 2 of the objects. So as can be seen, we switched the top 2. And so we would expect our control group to spend more time with these 2 objects, and remember that they were switched. And so the way we calculated this was through a recognition index, where we hand-timed the total amount of time the rat spent with the new object over the total time that the rat spent with the different objects. And then for the object in place, we hand-timed again how much time the rat spent with the 2 objects that were switched over the total time. So our results, so here on the graphs, we have the 3 groups. So we have the control group, and then we had 2 viruses which essentially have the same effect on these rats. And here is the recognition index. Although these results might seem promising, my mentor ran some statistics, and there is no significant difference between these 3 treatments. So the current work that I was doing, and actually finished recently, was running a T-maze test, which is actually another form of testing their working memory. And so in this test, we put the rat here in the middle, and then I put food pellet on each side. It's actually not cheese. And then it consists of a forced trial and a choice trial. So for the first forced trial, I blocked, let's say, the left arm, and then the rat went and ate the food pellet. And keep in mind, we train these rats. So before we ran these tests. So after the rat ate the food pellet on the right, we waited for 10 seconds, and then there was different delays. So there was a 10-second, there was a 70-second, and a 130-second wait. And then after that, we allowed the rat to go to either side, so we didn't block any arm. And so the rat should remember, quote unquote, that, hey, I ate the food pellet on the right, so why should you go to the left and eat the other pellet? So we are still in the process of running some statistics. And future direction includes some tests that test for depression-like behavior and anxiety-like behavior. So here on the left, there's a light-dark box task, which tests for anxiety-like behavior, and then the pour salt swim test, which tests for depression-like behavior. Having said that, I would like to thank UC Leeds for the great opportunity to do research. Specifically Erica, Maria, and Kevin for positive feedback. And then also the Salimsky Lab group, specifically Dr. Karen Salimsky and my lovely mentor, Sierra Love. Thank you.